Всем привет! Это Дмитрий Плешков, компания Technoinfo. Сегодня у нас в гостях основатель компании IVIM Technology, доктор Pilhan Kim. Hello, Dr. Kim.
Hello, Dimitri.
We did a roadshow with 7 seminars, and after this great week, what are your impressions about Russian scientific community, about Russian science?
Yeah, it's my first time to Russia, so I didn't have much idea what should I expect. But it was really a great week, and I met many Russian scientists, and then I received a really good impression, really great impression. They are all sincere, and then very very interesting, and then I think we can make a good collaboration in the future.
All this week we've been discussing intravital microscopy methods, intravital microscopy instrumentation. So please describe briefly what intravital microscopy is.
Okay, so intravital microscopy is a technique to image the cell in a live animal model in vivo. So it's based on two-photon and confocal laser scanning microscopy. But we can use this microscope to shine the laser beam into the live mouse model, and then we can take a very high resolution image of a live cell moving inside the animal model.
Sounds really great, and the presentations are so. It really looks like I never seen that before. So, is your equipment as unique like it look like?
Yes, it is. People have been trying to do intravital microscopy with conventional confocal microscope, but it's very difficult, because we have to take care of the live mouse model during the imaging, and then you also need to deal with the motion artifact due to the moving tissue inside the live mouse model during the imaging. And then our intravital microscope has a solution for all those challenges of intravital imaging. Okay.
So, but there are a lot of challenges. What is the key? So first, moving animal. Yeah. So you have motion compensation system. How it works?
Okay, there are two parts in the beginning. So our intravital microscope has a lot of accessory to stabilize the motion of the internal organ in a live mouse model, either by using mechanical force like negative air pressure or some pressing method with the holders. And then in addition to that, we also have an automatic image processing algorithm. It's capable of automatic image registration. By doing that, we can remove the motion artifact in our image in our intravital imaging data.
Does it work? Does it make the problem disappear completely?
Yes, it did. So I have been worked on intravital microscopy for almost 20 years. So I have a lot of experience to image all different organs in a live mouse model. So we have solutions for almost every organ to remove this motion artifact from the imaging.
Okay, so do you think this is the key feature or something else makes your system so good?
Yeah, this is there are many key features, but I think this is one of the most important key features of our intravital microscope.
Okay, so we found the solution for the moving animal. Mm-hmm. What else? We need anesthesia. You have one, right?
Yeah, of course. We can use any conventional anesthesia with our intravital microscope. We can use injectable anesthesia, and then we can also use gas anesthesia as well, like isoprene. We have a solution and accessories to use all those conventional anesthesia with our microscope.
What about other conditions for the animal? So you have heating stage.
Oh, yes.
What else?
Mm-hmm. Heating stage is very important because once the mouse is anesthetized, their body temperature drops very quickly. So it's important to keep their body temperature to be stable during the intravitreal imaging for several hours. And then we also have a solution for that. And then we have an all-in-one designed system in our microscope. So we integrate heating system and the body temperature monitoring system, and they all comes together into our intravitreal microscope, and then it makes the mouse to be alive and the body temperature to be stable, and then anesthetized for a longer period of time and stabilize the imaging condition.
And what is I found out is very important is all these, I don't know how to tell it, devices, small devices that you put on the mouse to stabilize and to fix the area that you are imaging.
Yes.
Do you develop them by yourself or you buy them somewhere?
We have developed this imaging accessories to stabilize the motion of the tissue by ourselves. So we make it by ourselves.
Great.
And then we optimize it with our microscope.
So you have
as a total solution. Sorry.
Yeah. So you have a huge experience in this area. So what is your background? And I know you're working in university.
My original background is electrical engineering. Okay. So and then I received a PhD in electrical engineering. So at that time, my topic was a telecommunication system. But I moved to this intravitreal microscopy in biology when I did postdoc training in the United States. It's more than now 20 years ago. So I switched from electrical engineering to biology and then start to develop this intravitreal microscope there from the scratch.
From the scratch?
Yeah.
Do you have any competitors on the market?
I think no, because this intravitreal to make intravitreal microscope to be optimal, it's important to understand the hardware itself in the beginning. Okay, so we need to know how to optimize the microscope in terms of optics, electronics, and mechanics. In addition to that, we need to understand the biology as well. Okay, because we are dealing with the live animal model, so we need to understand the biology and how to handle the animal, and how to stabilize the motion of the mouse model, how to maintain their body temperature, and so on. No other company has those broad experience in terms of hardware, software, and physics, biology, and optics. And I think the IVIM Technology is the only company that can do all those things together.
How many systems you already sold all over the world?
We already sold more than 25 systems globally. So in Korea, United States, China, and other countries.
Not bad for startup. You're
Yes.
a kind of startup company, right?
Yes.
So what is the history? How did you came up with the idea of making this unique instrument?
I started to work as a professor in the university, and then we developed this intravitreal microscopy technique, and then we want to commercialize our technique, okay, and then make it more accessible to the researchers in all over the world, right? So we started. I founded IVIM Technology at 2017. Okay, so at that time we received investment from the venture capital, to make our prototype in the beginning. So we spent 1 year to make a prototype, and then we released our first generation model at 2018. And then from that, we just did everything. We did marketing, and then and then meeting the partners like the Technoinfo, and then do the marketing activity in global exhibitions, and then that's it.
Question about money. So it's expensive systems.
Yes.
You need some money to start. So you mentioned venture funding. How did you get it? So you was looking for people with money and asking, look, I have a great idea, please help me. How it worked?
Yeah, more like that way actually. So South Korea is investing a lot on science and technology. So like Silicon Valley, now there are many venture funding capital in South Korea. So when I decided to start the company, I just asked to my colleague to introduce me to the venture capital. And then I did a lot of a meeting to persuade them, you need to give money to me to start the company. So that's how we started.
It was kind of some person with money, or it's some organization maybe from Korean government that accumulates this funding to push the researchers?
Well, you can say like a bank, It's like a bank, it's pond, But
so fun.
It's a pond.
There are a lot of features that your system has, and one of the key components, I think, is two-photon laser, right? So you can use either conventional one, Titan Sapphire, or you call it compact one. So what is the difference and what is the advantage of the compact model?
Okay, so first one, the conventional titanium sapphire laser is very expensive, and then it takes a lot of money to maintain it. This new one, it's a new product in the market. It's based on the fiber technology, so it's much more compact, and then it doesn't cost any money to maintain it. First of all, it's very cheap, much cheaper than the conventional titanium sapphire laser, and much smaller, and then it doesn't cost to maintain. And then it's small and compact enough to put into our microscope box. So it makes our system to be very compact in overall.
So you have different types of the systems.
Yes.
I saw it on your presentations, on the
Sure.
brochure. What is the best seller? What is kind of best product?
Okay, the best product is dual mode confocal two-photon integrated intravital microscope model called CMS3. Okay, that's our main product. So it can do both of confocal imaging and two-photon imaging in a single box. So it's one of it's very unique microscope in the market.
And this one is using compact two-photon laser.
Yes, and then it has compact fiber femtosecond laser inside the box.
Okay, and with two-photon excitation, you can work with dyes, and you can also use second harmonic generation. And usually, I by myself sometimes don't think this way, but second harmonic generation gives you actually a lot of information.
Yeah, additional advantage, of course.
So, can you explain a bit how it works and what it could be used for?
Okay, of course. Okay, so two-photon microscope, in the first place, they can detect the fluorescence signal like a confocal microscope. But in addition to that, this two-photon microscope can detect optical signal, nonlinear optical signal called second harmonic generation. Okay? And then this second harmonic generation signal can be generated from certain molecule like collagen in the live tissue. So without any labeling, without any additional labeling like fluorescence, this second harmonic generation signal naturally generated from the collagen in the biological tissue. And the two-photon microscope can generate this second harmonic generation signal together with the fluorescence signal. Okay? And then, so as I explained, this second harmonic generation signal can be generated from the collagen, so we can image the collagen in the biological tissue in the live mouse model. So it's very useful to study certain disease model, like including fibrosis. So this fibrosis is a disease that accumulates collagen in the tissue, right? And then this two-photon microscope is capable of imaging those collagen in this fibrosis mouse model. And then in addition to that, it can also detect conventional fluorescence signal as well. So it gives you much more information.
How deep can you get into the tissue?
Depending on, okay, so it depends on the type of tissue. For example, in the brain, we can, by using two-photon microscope, we can penetrate up to 500 micrometer in the brain. But in case of skin or muscle, we can penetrate up to 250 to 300 micrometer.
Okay, so wow, we have second harmonic generation, we have two photon excitation, we have four lasers, quite a lot. Are there any features? What are actually the main features of your microscope?
Okay, our intravitreal microscope has many good features, of course. First one is it's compact, it's all-in-one system. So it has everything inside in a very compact box. So it's easy to install. And then everything is automated, so it's easy to use. And then it's a small size, so it's, we can save a lot of space and cost. And then it can install it anywhere in the lab because it doesn't need a dark room to operate. It also has a very high-speed imaging capability. So it can do real-time imaging with a speed of 30 frames per second. Using this fast imaging capability, we can compensate the tissue motion with the image registration algorithm. And then everything can be done in automatic.
Good. Great. But as I know, you can speed up to 100
Yes.
frames per second. But you developed a special scanner for that, right?
Oh, okay. Yeah. So that's another unique feature of our microscope compared to other microscope company. We use a special scanner called rotating polygon scanner. It can rotate ultra fastly. So that's the key component to implement this high speed of 30 frames per second to 100 frames per second maximum.
Who is your main user?
Okay, so as I told you, we already sold more than 25 systems globally. So our main user is researchers working in university and then research hospital and also pharmaceutical companies.
Okay, and what is the main application?
Oh, okay. So their main application is to study the disease animal model. Also, they want to test their drug in the mouse model. So we can image the cell. In addition to that, we can image the drug in a mouse model by using intravitreal microscope. So it can be used to study the disease mouse model, and then it also can be used to test the drug delivery and drug efficacy in a live mouse model. So that's why this pharmaceutical company and research hospital want to buy the intravitreal microscope.
So you also sell to pharmaceutical companies?
Yes, we do.
Are there any big brands?
Yeah, we recently sold one system to Sanofi. Intravitreal microscopy can apply to many different topics in biology. So it has been, so our customer have used it to study tumor in oncology and also immunology and stem cell biology.
Three main.
And then also neuroscience as well.
You are making system really simple, but it still sounds a bit complicated. So especially if you're talking about sample preparation. So do you help researchers who get your instrument to start?
Okay, well, it can look complicated in the beginning, but we provide the very detailed training program for our customer to learn how to handle the animal model and then how to do surgery in an animal model for intravitreal imaging. So it's not too much complicated. After this training, normally our customer has been able to do intravitreal imaging experiment with a live mouse model within a couple of weeks. Okay,
so and how long is this training typically?
Typically it takes 2 to 3 days.
Only?
Yeah, and then it's practice.
Okay, but if they want more, can they come to Korea?
Of course. Yeah, we continuously provide the training service for our customers. So if they want to have a new surgery training, then we can provide that. If you ask about the potential of this intravitreal microscope, so as you know, now many people use IVIS system to do in vivo imaging.
IVIS,
yes. But it's a low resolution macroscopic imaging. But it's now everywhere, so everyone is using this, IVIS system to study the tumor, oncology and immunology and so on. And then now it's time to make another, our imaging to another level in several resolution in vivo mouse model. So intravital microscopy is a key breakthrough technology that enable to obtain this higher resolution cellular level in vivo imaging in a mouse model. So it can be a good synergistic system complementary to the IVIS system. So I believe in next 5 to 10 years, the needs for the intravital microscopy will go very high. And then everybody want to use this intravital microscope together with the IVIS system.
Okay. Can I buy stocks of your company at this stage?
Yeah, of course, actually. Our stocks will be in Korean Stock Exchange in maybe next year. Yeah.
Really?
Yes.
Oh, great. Okay. I will think how to make a bank account in Korean Stock Exchange. Mm-hmm. So you have a great future. Are you prepared? Do you have in your mind some new features or maybe new instruments, new major upgrades?
Yes, we are preparing a major upgrade next year. So now our model is a third generation model, but we will release the fourth generation model next year. It will have more desirable feature like improved stability, and then and also additional options, customization option like endoscopy imaging, and also retina imaging, and also other challenging organ imaging like heart imaging, for example. Okay, so you will have more features, and then you will also have an additional feature like a stimulation system as well. And then there will be also major upgrade in our software as well. Okay, so we will include a lot of machine learning based AI toolkit to our imaging software. Yeah. So it will be more automated, and then our users will be able to do a lot of nice image processing like denoising, for example, and then segmentation, automatic segmentation and analysis.
It was a great talk. Thank you very much, Dr. Kim. And the system is awesome. The method is it's really breakthrough, I think. And can't wait to see you here in Moscow again. Mm-hmm.
Sure, yeah, definitely. Yeah, it was my first time in Russia. And then I strongly believe I will be again in Moscow in the near future.